Thanks for joining, everyone, especially the people who woke up very, very early. That's incredibly impressive. I saw Mike Hurwitz there at 5:45. That's very good. But I'm incredibly excited for our first session of the day, which is really focused on immunotherapy. And one of the things we thought about for this session is really bringing people from outside of kidney cancer in to see what are some of the innovations happening sort of throughout. And so the hope is we can really learn from folks who are experts in gut microbiome, and obviously there's been huge developments from Nosley, from Monty and others, in PD-1 VEGF bispecifics, really from a company that started this field, looking beyond what are conventional antigens like neoantigens, some pioneering work in pancreatic cancer that we're going to hear about and maybe ties into kidney cancer as well. And then really kind of a new area, which is AI-guided protein design and really bringing in the larger sort of ecosystem of the Parker Institute. So really excited for today's session.
Good. The first speaker.
Perfect. So for our first speaker, we have Dr. Bertrand Routy, who really is one of the people who started this idea of the gut microbiome modulating immunotherapy responses. And we're so glad to have you join us today.
Great.
Thank you, Tony and Monty, for this nice invitation. I will talk, of course, on the gut microbiome, my disclosures. So really, this slide sort of summarizes 10 years of work and the major discovery that propelled this dark matter from one of the hallmarks of cancer and now randomized trial in the US. Very briefly, we demonstrated back in 2018 that kidney cancer patient that responded at a unique microbiome composition, and that led to really using the microbiome as a biomarker of response. More than now 90,000 patients within 100 trials. We demonstrated in the meta-analysis that is coming out that really antibiotic prior to immunotherapy decreased efficacy. And 2 years ago, ASCO wrote an educational guideline on antibiotic stewardship. With the help of Tony, we demonstrated also that MADCAM, so a soluble immune checkpoint in the gut, was prognostic for kidney cancer. But more importantly, for the clinician in the room, is all the different strategies that are currently being tested, and it will be the purpose and the focus of my talk, to improve the microbiome composition. And today, I will really mostly focus on diet and a new paper that just came out a week ago in Nature. So first and foremost is the fecal microbiota transplantation. So we take the feces of a patient that responded or a healthy volunteer. And this has been actually performed and published in more than 10 studies in the refractory and the first line setting. And all these trials have shown first safety, but more importantly, very encouraging clinical signal. With respect to renal cell carcinoma, my good friend Yaniro in Rome published the largest randomized trial when randomized a patient with advanced renal cell carcinoma to a pembroaxetinib plus or minus FMT from two kidney cancer that had experienced extraordinary response. Even if the study did not meet the primary endpoint, you see clearly a very nice difference in the PFS graph with 24 months of PFS in the FMT group compared to 9 months in the placebo plus immunotherapy group. Of course, one of the questions is how transferring bacteria from a donor to a patient could improve immunotherapy response. Our first hypothesis, and the first graph is from our melanoma and non-small cell lung cancer patient, we of course hypothesize that, okay, we transfer good bacteria to the patients, therefore these good bacteria will engraft and will increase immunotherapy efficacy. And in two papers, the first one in 2023 and the last one from January, in those papers we're quite surprised to see that the number of bacteria that patients received from the donors were equal in both responder and non-responder. So the absolute engraftment rate is not the mechanism of this efficacy. Going back, we also look at the bacteria that were shifted and that were lost from the patients between the responder and the non-responder. And you see in the first graph, the FMT illuminate, in non-small cell lung cancer, the responders seem to lose more bacteria. We then went back to the three first FMT trial, and once again we realized that the responder patients were actually the ones that were losing more bacteria after they got their FMT. So of course we reached out to Gianluca in Rome, and we asked him, in your randomized trial, can you also look at the number of bacteria that were lost in the responder versus the non-responder after the FMT. And guess what? He also demonstrated in his randomized trial that only in the FMT group, the responders were the ones that were losing bacteria. Of course, which one are these bacteria? And in my introduction, I told you we identified good bacteria, but the field is really moving towards the bad bacteria that are enriched in non-responder. And interestingly, when we look at the top 50 bacteria that were lost by the responder, we see that most of them are these deleterious immunosuppressive bacteria, Enterocluster and Clostridium. So this really is the first demonstration of the mechanism by which FMT eliminate deleterious bacteria to improve immunotherapy response. Despite my excitement, FMT is very challenging and clearly not scalable, so we have to find a new option. So in the States, you have the chance to have Montipal and Nasli, who have been working on this field. So instead of giving, and Nasli, you arrived just for your picture, perfect timing. So instead of giving the entire feces like we do in Canada, in the United States, of course, they're more elegant, and they give just one bacteria, Clostridium butyricum. And they published two wonderful studies, again, the same clinical design, 2-to-1, the first trial with Ipi-Nivo, the second Nivo-Cabo, and the patients were randomized to the immunotherapy plus or minus the CBM. In the both trials, surprisingly, the primary endpoint, which was a microbiome endpoint, was not met. They did not demonstrate that these bacteria significantly changed the microbiome. Nevertheless, when you look at the clinical outcome, the partial response rate in the Nivo EP CBM was 58%, significantly higher than EP Nivo alone. And in the Nivo Cabo, 74% achieved partial response, so that's quite extraordinary. And thanks to these two champions, now the SWOG has launched. I was told already 10 patients have been enrolled in this large SWOG trial where patients with metastatic renal cell carcinoma will receive IO-based therapy chosen by the investigator and will then be randomized to receive a CBM or placebo. And the primary endpoint will be PFS. So beyond bacteria, there's also prebiotics. So prebiotics are small molecules that have the potential to improve your microbiome. Back in 2022, we demonstrated that a Brazilian berry called camu-camu had an active ingredient, a polyphenol called castellagin. And in murine model, we showed that castellagin could have a synergetic effect with immunotherapy. This is melanoma, so refractory melanoma patients. So we launched and completed a trial of 15 patients refractory to immunotherapy, to BRAF inhibitor, were then re-challenged with immunotherapy and camu-camu. And 3 out of 15 patients responded very well, with 1 patient in complete response, and 2 had very sustained and prolonged disease. And again, a reminder for the non-melanoma expert, this was either palliative care or entering in this trial. Interestingly, the effect of this prebiotic was not as dramatic as the FMT in terms of the microbiome, but was more subtle, but mostly in the metabolite. And my new obsession, and I don't have time for 10 minutes, but it's the bile acid, and we demonstrated that castellagin increased microbiome-derived bile acid and mostly ursodeoxycholic acid. Of course, it's a kidney meeting, and of course, Monty, when we talked about it, is this nice results, he got very excited. And he launched another trial with the same design, 2-to-1, Ipi-Nivo plus or minus camu-camu. And you see the camu-camu arm, the response is quite extraordinary with regards to PFS. And lastly, and I told you, the main focus of my talk will be the diet. So what ASCO tells us about the diet, they don't tell us much, because as you see, they say that there is insufficient evidence to recommend for or against any dietary intervention. So we did this large exercise to combine 5,000 serum of patients treated with immunotherapy, and among them there was a lot of renal cell carcinoma patients, including patients from Tony Checkmate 025. And using an ablation process and machine learning, we demonstrated that 3 factors were the most important on these patients who respond to immunotherapy. So high BMI, and we had a nice talk yesterday, and histidine. And for the kidney aficionado, again, in the checkmate 025, we measure histidine, and those with the MSK prognostic risk that were favorable had a higher level of histidine. And you see both PFS and OS were prognostic when we segregated with the histidine level. Of course, histidine is found in the diet, in white fish, and in Montreal, we are performing dietary survey, and this is the result of 147 patients with non-small cell lung cancer. And we were able to demonstrate that those that were eating more histidine were actually responding better to immunotherapy. Of course, then we say, okay, let's try it in mice. And you see on the right that if you give PD-1, it works very well in mice. If you give PD-1 plus histidine, it works even better. The challenge for future clinical trials that we say, let's increase the dose of histidine, and at a higher dose of histidine, it was not actually as potent as a lower dose of histidine. And lastly, this paper that was published last week in Nature, so we sort of investigated 12 different diets, and we had really some unexpected results. So if I were to ask you, among these 12 diets, if you're a kidney patient, which diet would you prefer that Monty or Tony or Nasli recommend you? Psyllium, so you can beautiful, nice seeds, Mediterranean diet. There's a lot of paper from Jennifer Wargo's group that show that Mediterranean diet is good for melanoma. Japanese diet, potentially ketogenic diet, like looks very good on the picture. Vegan, everybody should be vegan or told to be vegan. However, when we look at more American, and don't want to say who encouraged this type of diet in your country, but Western, American, aspartame, of course, everybody wants to stay away from that, as well as high-fat diet. So with my colleague, Daniela Quell, we tested all these diets in mice. And quite surprisingly, the best diet, so the Mediterranean, the Psyllium diet, the Japanese diet, were actually the worst diet for the mice. While your country's diet, American, aspartame, high-fat diet, were actually the best. So I'm not going to tell you to follow your government recommendation because there's a twist. So first of all, we look at the microbiome of this diet, and interestingly, these obesogenic diets were actually good for the mice microbiome because they increase our favorite bacteria, Akkermansia and Lactobacillus. And when we look at the flow cytometry of the mice that received this obesogenic diet, they were also the best. They were more T cells, less neutrophil, and we increase in the non-responder mice the PD-1 Ki67 negative, so non-activated CD4 and CD8 T cell. Then more importantly, we performed metagenomic on these mice, and we found one bacteria, Lactobacillus johnsoni, that was increased in this obesogenic diet. Again, to tell you that the future of oncology is not the American diet and to become obese, we also tried to give the diet for only 3 weeks and not 15 weeks. So if you give the diet for 3 weeks, the mice do not gain weight. Their microbiome shifts as much as the 15-week diet, but they do not gain weight. And you see the Psyllium, so the diet that does not work for 15 weeks, at 3 weeks it does not work also. And the high-fat diet that works at 15 weeks also works at 3 weeks, and the mice have not gained weight yet. And we showed a similar result from an immunological standpoint. So then we wanted to show that a short course intervention was sufficient, and you did not need to gain weight. So if you start with the bad diet, the Psyllium diet for 21 days, you put the tumor, and then you switch rapidly to the high-fat diet, the good diet, you see that PD-1 works very well. Conversely, when you take the high-fat diet, so that works very well, you give it for 21 days, then you switch to the Psyllium, so the bad diet, you see that you inhibit anti-PD-1 efficacy. And similarly, we demonstrated that CD8 TNF production was only good when you started with the bad, but you switched with the good diet. We then, of course, perform FMT from non-responder patients, and if we gave the bad diet for 3 weeks, we were not able to restore anti-PD-1 efficacy, but if we give the good diet in this non-responder patient, we're able to transform. And again, to make sure that you understand the conclusion, it's not to be obese, it's not to eat McDonald's every day, but it's maybe to eat the good bacteria. When we give the good bacteria, the Lactobacillus johnsoni that was increased in this obesogenic diet, we give it to the bad diet, the psyllium diet, we're able to restore anti-PD-1 activity. And if you are on a high-fat diet, these are the mice, of course, that have not gained weight, and we give this Lactobacillus johnsoni, we actually cure the mice. Then, of course, beyond the diet is the metabolite, and we demonstrate that aldol acetic acid was increased in the mice that received this high-fat diet. And nicely, we demonstrated in non-small cell lung cancer that the same metabolite was present in responder. So in conclusion, so the field is moving fast. Nevertheless, I think, and this is Tony's recommendation when I asked for advice, that Bertrand, you need to show me a large randomized trials to confirm that what you do is useful. Otherwise, bye-bye microbiome. So I'm very happy that in the US, the SWOG are doing this large randomized trial. In Canada, where we launch and enroll half of 128 melanoma patients in the largest FMT randomized trial. And I hope over the next few years we will have large randomized trial to confirm or not the importance of the microbiome. And very quickly, so FMT is safe, 10 positive trials, however it's not scalable, and we identified the mechanism of action. Castelagin, it's encouraging. We understand it works through the bile acid pathway. LBP, so there's a lot of trials that are ongoing. FPRO, potentially Akkermansia trial, and in the SWOG trial. And of course for the diet, so this is unexpected that the obesogenic diet improve IO response. And we sort of understand the paradox of this in immuno-oncology. However, a kind reminder, obesity is not required for you to respond to immunotherapy, but to have a good microbiome is key to respond to immunotherapy. So on this, of course, I want to thank all my collaborators, my partner in this journey, Dr. Al-Kriev, Daniela and Logal Wash on this beautiful Nature paper, and my good friend Monty and Nazli, who are leading microbiome research in the US, and all of you. Thank you very much.
Maybe we'll take some questions, actually. Do some questions. I have some questions for Adam.
Hi. Sorry, I don't know where the mic is.
We're here.
Thank you. Hi, Adam Dicker, Jefferson. So thank you. Could you comment upon the precision oncology pre-treatment versus, and then how to pick the right microbiome strategy?
That's a great question. So we know that 50% of patients respond to immunotherapy without any further intervention. So of course, the goal will be, and future clinical trial will be to have access to rapid sequencing, which we're doing, using different scores such as TOPO-score, and then you screen the patient based on their baseline microbiome composition, MADCAM level. However, unfortunately, in 2026, I'm not sure we're ready for this because it will take time. The signature, there's delays in metagenomics, so we're working on developing qPCR assays to overcome this. And also, if you select for patient population, then enrollment rate is actually quite slow, and we don't fully understand yet exactly the specific signature. So that's really the future, but before thinking of tailored microbiome intervention, we have to try, see if it works, if it does not work, and why certain patients work, why certain patient does not work. So I can tell you, for our Canadian randomized melanoma trial, we're prospectively following this engraftment rate. And of course, if the trial works or not, we could sort of propose then another trial where if there's not enough bacterial loss, we could sort of do a salvage FMT or another intervention.
Okay.
I wanted to ask if you've been able to show, as patients get resistant to immunotherapy, if the microbiome actually changes and the bad bacteria become predominant. Have you been able to show anything like that?
So unfortunately not. There's a lot of effort, especially at Gustavo Roussy, to try to answer this question. We have a paper coming along that Immunotherapy does not change the microbiome over time. Even chemoimmunotherapy, so I do mostly lung, does not impact significantly the microbiome. And then, of course, the dream, so that's the first story, that IO plus combination does not change significantly. And of course, to use the microbiome as sort of ctDNA, that's my dream. But to collect patients at the time of progression is challenging, it will take time, but Nazli, that's a nice project for you to start.
Phenomenal presentation, great data. A quick question for you, and I'm surprised you and I haven't chatted about this before, but Nazli and I and Hedye through our studies, we're sitting on pages and pages of diet logs from our patients. And at this point, do you have any perspective on what the best methodology is to capture diet-related data, what questionnaires we should be using, et cetera?
It's a very good question and very challenging. So we use the NIH dietary survey, and we just published a paper in Gut Microbiome where we demonstrated in 150 Canadians that unlike MD Anderson data, there was no correlation with high and low fiber. And then to demonstrate that actually the Canadian patients were eating very poorly, I asked everybody in the lab to fill up the dietary survey, including myself. And it's very difficult to fill it up, because if I ask you what's a portion of vegetable, what's a portion of meat, if there's not a nutritionist next to you who tells you this is one portion, you will fail. And me, the first time I did the test, my fiber level was very low. Then I asked the nutritionist if there was something wrong. I ate well, and then I sat with her, I did the test with her, and then my fiber level sort of doubled. So I think that's one major challenge. The second challenge is that I'm still convinced that a steak in Houston, a steak in Paris, and steak in Burgundy, a steak in South Africa, is it the same meat? Is it the same steak? If you like it medium, or probably you overcook, versus me, I like it rare. What's the impact? So this, I think, is unknown. And the biggest challenge, and we put that, and if they have time to tell you, in the Nature Medicine on histidine, is of course one smart reviewer asked us, okay, you show that patients that eat histidine do better. Show me that if you eat histidine, you have more histidine in the blood. And we're not able to demonstrate that. We were actually able to demonstrate that those that eat more histidine, that have a good microbiome, have more histidine. And if you eat a lot of histidine, but you have a bad microbiome, you metabolize the histidine immunosuppressive metabolite. So it's very challenging, and we don't know yet much.
One more question.
One last question.
Maybe Kelly?
Hi, Dr. Rowley, huge fan of your work. How do you see the field moving in the next 5-10 years? Are we going to be prescribing our patients take these histidine tablets, or eat 30 grams of fiber a day, or are we going to find how they actually, at molecular level, how they impact the tumor microenvironment, and then target the targets that they use?
Yes, I think that the big elephant in the room is what is the mechanism. I hope that the field, and me, I'm worried that if we do intervention without understanding what they do, we will fail. And of course, we have to understand what it does to the microbiome, to the metabolite, to the immune response. With your SWOG trial, I think you have the opportunity to answer a fantastic question. And again, that's why for those who will put patients on this trial, please collect sample, collect serum, collect plasma, and feces, because it's the only way we will be able to show. I hope that in the future we will have, so to answer the first question, and we're working on it, so quick qPCR turnaround time to demonstrate good or bad microbiome. Good microbiome, please proceed with your immunotherapy. Bad microbiome, horrible, you need the LBP 150 canvas versus, or it's a borderline, it's not perfect, okay, you need a bit of CBM, a bit of Castalagen. But again, I think that the field, and again, Tony is very critical in my work, that we need a large phase 3 randomized trial to convince mostly the oncologists that this is important. And then I think that the field will expand.
Outstanding, fantastic presentation. There will be more communication offline. The next speaker is Fiona Ahn from Summit Therapeutics. I would like to invite her to come up.
Good morning. Thank you very much for the invitation. I'm Fiona. I'm the Global Clinical Lead from Summit. Here's my disclosure. So ivonescimab is the first-in-class bispecifics. It targets both VEGF and the PD-1. So it brings two valid targets onto one molecule. So by binding PD-1, it blocks the interaction of PD-1 to both PD-L1 and the PD-L2. So by binding the VEGF, we all know that it's going to suppress the tumor angiogenesis. So how is this different from co-administration of a PD-1 monoclonal antibody and plus a VEGF monoclonal antibody? So the fundamental difference is that for these type of bispecifics, it has its unique cooperative binding ability. So what is the cooperative binding? So there are two folds of mechanisms behind it. Firstly, there are a lot of preclinical data done that showed that in the presence of VEGF, there are actually the increased binding affinity of ivonescimab to PD-1. So at least 18 folds of increase. And on the other hand, vice versa, at the presence of PD-1, there is an increased binding affinity to VEGF. So because it has a dual blockage of two targets, so in the tumor microenvironment, the both expression of both targets, so that will maximize the avidity of the drug in the tumor microenvironment. On the other fold of mechanism here is very interesting because VEGF actually forms the dimer. So because of the dimerization of VEGF, it's interconnecting each individual ivonescimab molecule. So as you can see on the right side on the graph, that the ivonescimab molecule actually interconnecting and forming this stable structure is like we call it daisy chain link structure. So what is doing that is really to localize the drug in the tumor microenvironment. So what does that do to the safety? So theoretically, because of the accumulation and the localization of the drug in the tumor microenvironment, and will limit some of the off-target effect to have better efficacy and to have better safety profiles. Another very important feature of this molecule is that the half-life is much shorter than bevacizumab. As you recall, bevacizumab has the half-life about 20 days. Ivonescimab has only 10 days of half-life. It's much shorter. So we believe this feature contributes to the manageable safety profile we have seen in the recent clinical trials. And finally, on the molecule, we have designed and engineered the FC null region. So that minimizes the FC receptor mediated the effector functions. So that will further reduce some of the immune-related side effects, which we have seen in the clinical studies. So currently there are very active, extensive clinical activities going on with ivonescimab. So Summit Therapeutics, we are in partnership with Akeso, and between the two companies, there are 15 global phase 3 or large regional phase 3 study, either completed or currently ongoing. So as you can see on the slide, so these studies across various different types of solid tumors, and including some of the traditionally we consider cold tumors, such as CRC or PDAC. So we're very excited to see some of the preliminary activities in these type of tumors as well. And we also have a lot of collaborations with our partners. We recently announced that we have a collaboration with our partner with Arcus, that we are going to investigate the combination with HIF2 alpha inhibitor. We also have the investigating the other combination with ADCs and with other IO agents as well. So the next few minutes, I'm going to go through some of the non-small cell lung cancer, the data very quickly. In the interest of time, I'm going to just flip through them very quickly. So Harmony 2 is a Castle-sponsored study. So Summit, we have a similar study called Harmony 7 currently enrolling patients. So Harmony 2 consists of all the Chinese patients, about 400 patients in the frontline non-small cell lung cancer patients. The patients are randomized 1-to-1 to receive ivonescimab monotherapy or pembrolizumab. So ivonescimab was given every 3 weeks, 20 milligram per kg, and the pembro was given at a flat dosing of 200 milligram. So I want to mention that the patients, including, so in this study, there were about 45% of patients are squamous non-small cell lung cancer patients. So in the study, the results are very exciting. So the median PFS has been improved from 5.8 months for pembrolizumab to 11.1 months for ivonescimab. So the hazard ratio is 0.51. And even more exciting is that we see this clinical improvement that not only in the non-squamous patient population, but also in the squamous lung cancer patients. And similarly, we see this improvement across different PD-L1 level. So as a reminder that historically that for the PD-L1 low patients, the monoclonal PD-1 antibody did not benefit these patients. So ivonescimab was able to demonstrate the clinical benefit in these patients. And even more impressive is that for the PD-L1 high, or the highest patients, so pembro already had very good efficacy. Ivonescimab was able to further improve these clinical outcomes. So what about safety? I think that's probably the more important discussion here for this audience. As expected, because of the added target of VEGF, there was reported a modest increase of treatment-related adverse events. There were 90% of TRAEs for the ivonescimab arm versus 82% for pembro. And similarly, for modest increase for the grade 3 TRAE and the serious TRAEs. What's reassuring is that, as you can see on the slides, that the rate of discontinuation and the rate of grade 5 events are very similar across the two arms. And for the treatment, for the immune-related adverse events, very, very similar between the two arms, and even the types of IREs are very similar. So finally, the VEGF-related adverse events. So we have observed that the most common VEGF-related adverse events was hypertension and the proteinuria. So grade 3 hypertension was 5% for ivonescimab arm versus 1% for the pembrolizumab arm. And for proteinuria was 3% versus 0% for the grade 3 and above for pembro. I wanted to mention that there was no major bleeding events, which is very reassuring for these patients, especially for the 45%, almost half the patients on the study were squamous patients. These patients have increased the bleeding risks. And also, as a note, that bevacizumab actually is contraindicated for the squamous patient population because of the elevated bleeding risk. So I believe that this is a very strong clinical proof that for this molecule, for the bispecific, has much better safety profile relatively to bevacizumab. So very quickly for Harmony 6, this is a combination therapy. It's also in non-small cell lung cancer patients newly diagnosed. And these are all squamous histology, and also including patients with PD-L1 actually including patients with no PD-1 expressed. So the patients are randomized to ivonescimab plus chemo, followed by ivonescimab, given up to 24 months, versus tislelizumab, which is another PD-1 monoclonal antibody plus chemo, followed by tislelizumab alone. So very similarly, this study has demonstrated a statistically significant improvement for both PFS and OS, and these improvements have seen across different PD-L1 levels. So similarly, for safety, similar to the previous studies, there was a modest increase of the TRAEs for the IVO arm, but as you can see on the slide, the grade 3 and above IRAEs are very similar between the two arms. And the rate of discontinuation and the grade 5 events are very similar. So because of the entire study, over 500 patients were squamous patients, so there we have seen a slightly increase of the hemoptysis on the study, but it's limited cases in terms of the serious adverse events. In the end, I believe I'm invited to this conference for us to talk about the application of ivonescimab as a novel PD-1 and VEGF bispecific. What is application in RCC? So RCC is heavily VEGF dependent, and there has been great successes for checkpoint inhibitors. So on this slide is a phase 2 study of ivonescimab monotherapy in the clear cell RCC. It's IVORY study led by Dr. Pablo Masal from MD Anderson. So this study is investigating the ivonescimab monotherapy in the second line setting. So either for the patients have failed the prior IO treatment, or for the patients who have failed the VEGF-related therapy. So this study is going to be able to answer a lot of questions. I know Dr. Masal has included a lot of correlative endpoints, and samples are being collected. And I'm also happy to share that this study almost finished the accrual, and we're all excited and looking forward to seeing the data from the study. And I also want to take this opportunity to give a shout to Dr. Paul and Dr. Yep for their study in neoadjuvant. So I know the study was recently activated and is in the process in enrolling the first patient. So it's a monotherapy neoadjuvant setting. So if you are interested to know more details, please talk to Dr. Paul and Dr. Yep. So with that, just to summarize it up, so I firmly believe that ivonescimab is fundamentally different from monoclonal PD-1 monoclonal antibody or VEGF-targeted monoclonal antibody. So we have seen the efficacy in the patient population, such as like no PD-L1 expressed patients, and in the squamous patient population. And most importantly, the safety profiles were very well tolerated. So we believe is an excellent combination partner with other agents. So finally, I'm extremely excited about the opportunity to move this agent into the RCC space. I look forward to working with many of you going forward to hopefully bring this ivonescimab to patients very soon. Thank you very much.
Great. Thank you so much, Dr. Ahn. Any questions? We have time for maybe like 2 questions.
I'm going to ask one just while the microphone is going. Just about crossing the blood-brain barrier, is there signs of activity in the brain?
Yeah, excellent question, David. So we actually had published the 35 patient data, was a combination of 2 early phase studies. So total of 35 patients. 7 out of 35 patients were monotherapy ivonescimab. The rest are the chemo combo. So we have seen over a third patients that have seen the intracranial responses, which was very exciting. We don't completely understand what was behind the mechanism of the responses. So we have seen no bleeding events for these patients. We also have a GBM study, which is the investigator study currently ongoing to investigate, and hopefully we'll understand the biological rationale behind the responses very soon.
Thank you.
Thank you.
Very nice talk.
Any question?
Yeah. Oh, thanks for your very nice talk. You mentioned that VEGF dimers are bound by the agent and maybe differentially from VEGF monomers. Is it because these VEGF dimers are immobilized in the TME either by receptor or extracellular matrix, or is it another mechanism? With regard to the VEGF dimers.
So you're asking.
Yeah.
So you're asking how it's fundamentally different from
Yes.
the monoclonal and the VEGF monoclonal antibody?
Right.
Is that your question?
Yes,
Sorry.
it is.
Yeah.
I think the dimer formation versus
Yeah.
monomer
Yeah.
formation. The dimer formation versus monomer.
Yeah.
Oh, okay. So I think the bevacizumab actually has the similar effect, the dimerization as well. So because of the VEGF has the dimerization. So that's not unique for this agent, for ivonescimab. I think what's unique is because of the dimerization of VEGF, it brings the PD-1 targets together. So basically, it brings the T cells and the tumor cells and basically concentrate the drug around the tumor microenvironment. I think that's my understanding that's what fundamentally is different, and that's how we see this. The safety is different, is that it is because of the half-life. I think that is a major difference.
Right. So the fundamental distribution of the drug is different. The fundamental distribution of the drug is different than 2 separate agents. This is what you're...
The distribution is different. Let me suggest that the distribution is different, the distribution.
Oh, so you're asking the drug distribution, right?
Yeah.
No, kind of like summarize it.
So so I think it's the drug is metabolized like similarly to other tetravalent bispecifics. So it's not excreted by the kidney, and it's not metabolized by liver.
Got it. One last question for myself, so to say. So squamous carcinoma in the lung, there's contraindication for bevacizumab, right? The fact that you're getting away with this VEGF agent would suggest to me that you're not as rapidly devascularizing these tumors and then causing the bleeding event. Do you think that this more modest effect may actually induce what we had all had hoped for with the VEGF TKI PD-1 combination to maybe like make these tumors not too hypoxic, but to allow for more infiltration of immune cells?
Yeah, it's a great question. I think again goes back to the half-life, right? Because I think the similarly to bevacizumab, the drug is given every 3 weeks, so every 21 days. So bevacizumab, the half-life is 20 days. So basically, it's a constant suppression of the tumor vasculature. But with the shorter half-life, I believe that allows the vasculature to recover, right? You know, especially the healthy tissue, the vasculature would have a chance to recover from this suppression of the VEGF. So I think that may explain the less bleeding events. But you know, all these are based on the hypothesis and some of the preclinical models, and we are, you know, as you can see that there are a lot of clinical activities going on. So we are collecting all the samples and actively understand that a lot of the scientific rationales behind these questions.
Outstanding. Thank you so much. Oh, yeah, no.
Here, please have a seat. And our next speaker, I'm very pleased to introduce Wilfried Pastor, who I've known for quite some time in the pancreatic cancer world, but is going to speak to us about some of his work on the dark cancer proteome and applications to kidney cancer. Thanks for joining.
Absolutely. And thanks so much for having me. Good morning, everyone, and a huge thank you to the organizers for this invitation to speak today. I'm really excited to talk about the cancer dark proteome, which I promise I'm going to introduce what that actually means in a second. Really is a novel source of tumor antigens. As David alluded to, I'm not a kidney cancer doctor, but I think this actually has a lot of implications for kidney cancer and numerous other tumor types as well. These are my disclosures. So I run a tumor immunology lab at Dana-Farber. We're really focused on what are the optimal tumor antigens for anti-tumor T cells. Now, of course, there's been decades of evidence suggesting that peptides bound to HLA class I can be targets for T cells from a variety of sources. These, of course, include cancer-associated viruses, canonical tumor-associated antigens, and then a wealth of evidence suggesting that somatic mutation encoded neoantigens can be recognized by T cells, including, of course, in RCC from David Braun and Tony Choueiri and Kathy Wu and others, demonstrating that personalized neoantigen vaccines can elicit robust anti-tumor T cell responses in RCC patients. Now, what I want to talk to you about for the remainder of this talk, though, is to say not to discount those classes of antigens whatsoever, but to really say that the antigen landscape is much broader and deeper than originally appreciated. Really, over the last several years, we've seen a wealth of data emerge that says cancer cells, multiple different types of cancer cells, can go awry, not just transcriptionally, but also translationally, leading to novel antigens derived from splice products, misincorporation of the incorrect amino acid in a stereotyped way in response to tumor-relevant stresses. We now know that across tumor types, a number of transposable elements are reactivated and can give rise to class I bound peptides that can be recognized by T cells. And even the ostensibly non-protein coding genome, these non-canonical open reading frames, it turns out that the translational machinery in cancer also goes haywire and can be recognized by T cells. Specific to kidney cancer, we saw some elegant work last year from Bill Kalin's lab and Kathy Wu's lab, really led by Qin-Qin Jiang and David Braun behind me, demonstrating that endogenous retroviruses can be directly transcriptionally upregulated by HIF in RCC. These can then be translated and give rise to peptides in the context of HLA class I and can be recognized by CD8 T cells in RCC patients. Now, my lab has historically not focused on kidney cancer. Instead, we've focused on pancreatic cancer and numerous other GI malignancies, and been really focused on this other class, these novel unannotated or non-canonical open reading frames. Just to set the stage here, these come from outside of the annotated protein coding genome and are thought to arise from aberrant translation of the 5' or 3' UTR, translation in alternative reading frames, these so-called IntOrf-derived open reading frames, and then as well as translation of long, putatively non-coding or link RNAs. Again, it turns out that these have coding potential under certain malignant contexts. To give you a sense of how we even think about discovering peptides derived from the cancer dark proteome, we, instead of using computational prediction, instead we turn to empirical antigen discovery. Now, this is showing what we do in pancreas cancer, but again, the same is applied to RCC and numerous other tumor types, where we can take bulk tumors, patient-derived cell lines, patient-derived organoids, xenografts, etc., subject these to extensive genomic and transcriptomic profiling, but most importantly, where we can immunoprecipitate HLA class 1 complexes, elute off peptides, and follow this with mass spec, a process called immunopeptidomics, to now directly identify what peptides are being presented by MHC class 1 or HLA class 1 in these tumor cells. And I'll just mention that all of the immunopeptidomics I'll talk about has really been led by Jen Abelin and Steve Carr at the Broad Institute. Now, when we did this in pancreas cancer, we were really excited to see that even in this low mutational burden cancer, we could empirically identify a subset of somatic mutation-encoded neoepitopes. This was very exciting to us. However, it was also quite humbling in the fact that a number of these tumor samples, we could find no evidence of somatic mutation-encoded neoepitopes, and even the ones where we could identify 1 or 2 somatic mutations per tumor sample. So we decided to cast a bit of a broader net. The timing worked out perfectly, that Kathy Wu, Aviv Regev, Claude Pro, and others were just starting to describe this novel class of non-canonical open reading frames in different cancer contexts. So we decided to augment our proteomic search space with these non-canonical open reading frames, and lo and behold, we found not just 1 or 2, but now tens to hundreds of these cryptic peptides in every patient sample that we looked at, and these fell into a number of different defined biotypes shown here. We were excited about this, but also we wanted to say, well, what is not known? And I try to be very careful not refer to these as neoantigens, partly because we don't know the true tumor specificity of this class. They're outside of the annotated protein coding genome, but what is the real evidence for tumor selectivity of these cryptic peptides? To directly answer that, we decided to take a translation-centric approach to this, thinking that these are thought to arise from aberrant translation of non-protein coding, the non-protein coding genome. We leveraged immunopeptidomics as well as ribosome sequencing to look at active translation, now across hundreds of healthy tissues, healthy organs, profiled from over 29 diverse organ systems. The first takeaway for us was that not all should be considered tumor specific. Some we could find evidence in 1 or more healthy tissues, suggesting that this is not non-canonical, but actually likely an error in how we're annotating the protein coding genome. But for about a third, a little over 500 of these non-canonical HLA-1 bound peptides, we could find 0 evidence of any translation, not just the peptide, but any translation of even the underlying open reading frame in any healthy tissue that we looked at. And we term these cancer-restricted cryptic peptides or cancer-restricted cryptic antigens. Now, excitingly, unlike most somatic mutations that are often private, many of these are often shared, particularly if we look at patients with the correct HLA restriction element. These were all Dana-Farber patients, and so we looked at HLA-A0201 and found that a subset of these cancer-restricted cryptic antigens were found in over 70% of our A2 positive patients, really suggesting that this could be a great class for off-the-shelf therapeutics, particularly if we could provoke a CD8 T-cell response against them. So to directly ask what is the real immunogenic potential of these, we decided to employ an ex vivo priming and expansion platform using HLA-matched healthy donor PBMCs that we then use to prime and expand autologous CD8 T cells. We've used this platform to now test a variety of antigenic, peptides from antigenic classes and compare this to more traditional somatic mutation-encoded neoantigens, which clearly are highly immunogenic. But our takeaway from this was that both ERV-derived cryptic antigens, which has been in close collaboration with Chin and Bill Kalin, and then our cancer-restricted non-canonical open reading frame-derived peptides are quite immunogenic, about on par in the same assays with what we see with somatic mutation encoded neoepitopes. But beyond that, this could also serve as a robust source for TCR discovery. So we developed this pipeline using DNA barcoded HLA class 1 tetramers paired with the 10x genomics pipeline to now identify full-length TCR alpha beta with paired antigen specificity. We've used this to discover a suite of TCRs against multiple antigenic classes ranging from mutations to non-canonical open reading frames to most recently ERV-derived peptides. And then we can take a subset of these and molecularly reconstruct these and use CRISPR-based TCR redirection, or TCRT, to actually create cryptic antigen-directed cellular therapies and actually ask the question, can these truly recognize endogenous levels of their cognate peptide on tumor cells? Using both in vitro and ex vivo assays, where we put these back in co-culture with patient-derived organoids, we could show that across multiple different cryptic antigens, we could elicit quite robust tumor recognition, but importantly, cytotoxicity. We also wanted to push proof of concept that we could do this in vivo, and using patient-derived xenografts from pancreatic cancer patients showed that cryptic antigen-directed TCRT could exhibit quite robust tumoricidal activity, although never eradicated the tumors in these models. Because we couldn't eradicate tumors, we started asking questions, well, what if targeting one cryptic antigen is good, what if we target multiple together? Can we preemptively overcome antigen escape variants? And that's exactly what we've seen to date. We've tested a variety of combinations of cryptic antigen-directed TCRs and seen that a number of these exert quite robust additive, if not synergistic, activity. And then just to tease at the very end, some of the work we've been doing with Chin, Bill Kalin, and now with Chris Garcia, and perhaps a perfect segue into John's next talk, we've been leveraging AI de novo protein design to move away from TCRs, but instead TCR mimic antibodies, formulating these as CAR T cells, and again showing now directly that these are sufficient to mediate recognition and killing of tumor cells expressing endogenous levels of their cognate cryptic antigens, including ERVs derived from kidney cancer cells. So let me end with just a quick summary. Hopefully I've convinced you that aberrant translation outside of the annotated protein coding genome is a frequent event in cancer, including in kidney cancer, and these can result in cryptic HLA-1 bound peptides. A subset of these cryptic antigens are often exquisitely restricted to cancer, shared across patients, and highly immunogenic. And then with just proof of concept demonstrated that cryptic antigen-directed cellular therapies, either TCRT or most recently TCR mimic CAR T, can actually recognize and kill tumor cells in vitro, ex vivo, and in vivo. And we believe these are now poised for clinical translation in the coming years. I've tried to thank the folks that actually did the work throughout the talk, but just to put a few faces to the names. The published work was really led by Zach Ely and Zach Kolstad, two former members of my group. Much of the current work was led by Gurjan Gunadine, a postdoc in my lab, in close collaboration with Chin Jiang and Bill Kalin. As I mentioned, all of the immunopeptidomics was made possible through collaborations with Jen Abelin and Steve Carr at the Broad Institute, in addition to many other collaborators that have contributed here, and then of course the funding sources that made all this possible. So thank you, and happy to take any questions.
We'll start on this side here, maybe Abhishek?
So I had a question about just the way the biology will probably pan out. Many of these are probably cryptic peptides are probably coming from heterochromatin regions that are opening up apparently, would be my guess. Have you looked at these regions probably are very stochastically going to keep getting shut down and not impact the biology of the tumor?
Yeah.
So thoughts on how the presentation of these cryptic peptides will remain as a consistent target?
Wonderful question. And I think, we're talking about the cancer dark proteome as one class. It really is many different classes, and some are clearly from epigenetically regulated. I think many of the transposable elements, ERV, is clearly regulated in that way. In terms of some of the non-canonical open reading frames, these are actually from aberrant translation of constitutively expressed mRNAs. And there's some emerging evidence, not that we have, but from John Prenzer and others, showing that a subset of these actually exert pro-fitness effect in cancer cells. We don't know why, we don't know what they're doing, but it turns out if you knock these out, the cancer cells actually don't like it, suggesting that there may be pressure to not turn off translation in cancer cells, although again, that's still very early days.
So that's just very quick follow. The experiment very specifically only targeting the aberrant translational product without impacting the canonical open reading?
That's correct, yeah.
Next question. Maybe Katsu here.
Thank you. Thank you very much for great. I'm very interested in antigen. If you look at the single cell data, for example, there are so many antigen, but T cell is usually dominated, like 30%, 40% of T cell is from the same T cell. That's let me think about that. How much these of antigen are actually kind of the T cell recognize and attack that antigen? I don't know the expression level of antigen, or I'm not sure, but do you think these cryptic antigen actually work as a real neoantigen in before the T cell treatment, for example?
Yeah. There's like 3 questions in there, so I'll try to answer. So again, I don't want to call them neo, right? They're genetically encoded in every cell in the human body. So again, we call them tumor selective for that reason. There is clear evidence to suggest that a subset of these can provoke an endogenous T cell response. You can find TCRs that are specific to these even in patients, but many of them have not been tested, and so we don't know that all provoke an endogenous T cell response. Actually, in pancreas, there's a lot of evidence to suggest that there are defects in T cell priming more broadly, that there may be hindrances in provoking an endogenous T cell response. That being said, a lot more work needs to be done to characterize, are these actually tumor rejection antigens in people, and what do they look like in terms of the endogenous T cell response? Much more to come in the future years.
Next question.
By all means, yeah. I can hear you.
Mike.
Thanks. This is a simplistic question. Have you looked at heterogeneity of these expression of these things within the tumor?
We are actively looking. So the short answer is no. The longer answer is we're trying. There's no such thing as single cell immunopeptidomics yet, but we're actively trying to do that.
Other questions? Okay.
Nope.
Good. So yeah.
Just a
I'll
quick
take one
thing.
more. I can't say
all
no.
about the T cells, etc. But what's the effect this cryptic antigen, maybe neoantigen, have on the rest of the immune system? The B cell, the tumor microenvironment, these Tregs, the macrophages.
Yeah.
Are we just scratching the surface?
Yeah, so the short answer is we have no idea. And so again, this is going to be an active area of investigation in the years to come. Love it.
Perfect. Last question from Ari, and then we'll move on.
Oh, please. Yeah.
I was more along the lines of some of the provocative work from Kaylin and from others on the role of HIF in regulating ERBs. I assume you're looking at this as well, but mechanisms for exposing these ORFs and your thoughts on the effects of drugs on actually promoting.
Yeah, 100%. In terms of some of the HIF-regulated ERVs can be upregulated with HIF stabilizers. We've actually shown that now in, didn't mention it for the 10-minute talk, but that this is applicable to other tumor types, including pancreas, but where we can expose these to hypoxia and upregulate many of the same endogenous retroviruses that were seen by Bill, Kathy, and others.
Do you think some of the other non-canonicals are also HIF regulated, or?
So we have no evidence that they're HIF regulated. We're actively testing a number of perturbations, tumor-relevant stresses to try to figure out what is the mechanism there, but we don't have the answer yet.
That's great. Thank you so much.
Outstanding. Thank you so much. So last but certainly not least in this session is with Dr. John Connolly from the Parker Institute. He's the Chief Scientific Officer. We'll talk about AI protein design. Thank you very much.
Thank you. Thank you very much. Thank the organizers and thank Will for that great introduction on AI-based protein design. So I think I just want to cover just a little bit in this space. It's such a rich area, but the area that we want to focus on today, is, as you've just heard, is some T-cell receptor design, particularly massive breakthroughs in the development of this space. And so just some background, the Parker Institute is a 14-center network across the U.S. We're funding about 1,000 projects in both clinical space and research. But one of the most exciting areas really is this AI-based protein design and the changes that are happening. And I think from the fundamental research side, we want to kind of give you guys an idea of where we're looking in the next 5 years and the research programs that we're funding and pushing and the big challenges that we have. There's a lot of challenges out there, but I think, as you've heard from Will, one of the biggest challenges is really target discovery. What's out there to actually target in this space that we can go after? And so, 90% of the targets that are out there and an increasing and growing list of antigens are really intracellular antigens that really require this processing and presentation in terms of class 1 and class 2. And so by harnessing mechanism to actually go after these and to identify and target them, this is really where we're going to be able to redirect many of the therapies that we have out there today, some of the incredible effector molecules that will give us these long-term durable responses. And so, of course, naturally, as we've heard and as you guys understand, the way the body does this is really through T-cell receptor engagement. And when you think about immunotherapy efficacy in the context of solid tumors, all really long-term durable responses have at their heart T-cell receptor engagement. That's the recognition event that happens in the context of checkpoint blockade, cytokine, targeted adjuvants, you name it. All of this ultimately induces, in the solid tumor case, a T-cell response, and often a broad T-cell response that recognizes intracellular antigens and drives this long-term durable responses that are out there. And so what we're hoping to, and this, of course, this leads to some phenomenal therapeutics that are out there, both TCR mimetics, and in the case of, say, synovial sarcoma, and you guys have seen, this is the phase 2 data of the Adaptimmune trial, just demonstrating that when you have a response and you can really hook into this, you can drive wonderful long-term durability. Now, there's a downside to this, and Adaptimmune is probably a very good example, that it takes a long time to actually identify these T-cell receptors. It's quite a workload to identify checkpoint responders, pull in the T-cell receptors, and then begin to de-orphanize them and identify what they actually interact with, develop them into something with drug-like properties. So it's not something naturally T-cell receptors have, improving their affinity, and then, of course, assessing their cross-reactivity. And so this is really where we threw out the gauntlet a while ago now in the AI space, is just saying as large language models develop and we begin to create hundreds of millions of these interactions between T-cell receptors and MHC peptide complexes, can we create transformer-based large language models that begin to deconvolve that? And this, of course, really broke out in a series of back-to-back papers in 2025 that really built those early transformer-based models, and really led by 3 different groups. This Jenkins group in Denmark, David Baker's team at the Institute for Protein Design, and Chris Garcia's group at Stanford University. And so what they did is they really took a look at, can we begin to build these transformer-based models to understand how T-cell receptors recognize these MHC peptide complexes, and then come up with predictive and, in fact, de novo designed molecules that actually do this. And so this really, those early transformer-based models that were initiated really in 25 led rapidly to the application of David's work in the RF diffusion space. So this is a model, RF diffusion, that is used kind of throughout protein design. There are other models that are out there, but it's one that really push forward. And then it allows you then to sort of to denoise the space and identify shapes that fit these different MHC peptide complexes. So you can the model can then set up a set of rules and then bind those shapes. You can then move this forward into the peptide MPMN, which basically finds amino acids that then fit those shapes. And then very quickly you can generate peptides or mini proteins, circular peptides, mini proteins, and in fact full-size T-cell receptors that actually interact. And so these then can be benchmarked iteratively, both in silico up against, say, AlphaFold 3, or other models that are out there, most recently from NVIDIA and the ARC Institute, to really identify things that truly interact. And that's all before moving into wet lab space. And so very quickly, we can use these models to develop something that's actually pretty interesting out there. And you can sort of chain these with your agentic AI. You can do this very quickly, and people are doing this now just over a very short period of time to develop candidate molecules that are then screened by surface plasmon resonance. And so this is, I think, some of the biggest breakthroughs here are really in the democratization of these designs, in addition to their ability to really accurately identify T-cell receptors. And so if you then go and look at your surface plasmon resonance, you can do killing assays, and you look at the affinity of these, you really find some remarkable results that are coming out of this, at least as good as what we're seeing from fishing out TCRs from checkpoint experienced individuals. One is really this is this idea of wonderful fit, really tight fit molecules that interact well. The other is we can use these models to affinity tune the T-cell receptors. So one of the things that they don't tell you is that T-cell receptors are naturally pretty low affinity. They normally bind by avidity, that there's lots of T-cell receptors on the surface of the cell. It interacts and identifies different MHC class I molecules that then triggers the underlying biology. If we want to make real kind of drugs that are targeting the space, things like TCR mimetic proteins that can be sort of mass produced, you really need to improve the affinity. And in doing so, you can actually get a lot of off-target cross-reactivity. So we're able to use the AI to optimize these and the large language models to begin to optimize these in silico and really move this down. And to the point where we can get things like, and I'll show you some examples, single amino acid specificity. So even, it's able to differentiate even single amino acids, which is absolutely essential in these pipelines as we move forward. And so I think these were all wonderful, and this is really state of the art now. There are different models, and I think recently, even as early as yesterday, that just came out, that were phenomenal changes and improvements in the efficiency of MHC peptide complex at TCR design. But it kind of highlights right now that we've been able to unlock kind of this undruggable space, this intracellular proteome. We can do this in a way that is really, really fast, but I think it's even more importantly, again, this idea of democratization, that anyone can set this up, and I'll talk about a couple of online websites where you can just sort of circle or put your peptide in and it'll design it for you. And then finally, it's sort of the clinical reality, the ability to increase the speed from years, as it took Adaptimmune to identify and optimize their TCRs, now to a matter of weeks where you could easily generate that molecule. And in doing so, you can de-risk it. There are still some challenges out there, though. And in fact, as excited as I am about this AI-based protein design in the TCR space, it's really the application in the de-risking space that I think is even more important and growing out there. So if you look at the major challenges in the TCR space, one is that we really haven't had a lot of success identifying new T-cell receptors that are out there. There's great work from many groups, and you've just heard from Will, some work to really identify these new antigens, but really we've been stuck with the same kind of tumor-associated antigens that are across many tumor types. So what we need to do is try to find ways to use AI to actually identify novel antigens, fish those T-cell receptors out, and in doing so, improve their specificity so that improve the affinity as well as the specificity. And I think that that's a huge challenge. And it's one that really has been tackled by a number of different groups. I'll talk a little bit about the work from the Parker Institute with Chris Garcia's team, but I just want to highlight Michael Birnbaum, Steve Elledge, others that are out there really trying to solve these problems of how we're able to not just identify high affinity T-cell receptors, but make sure that they're specific for their target and not for any of the other myriad targets that are out there in the MHC peptide complex space. And so Chris's approach was really using a yeast display system, and we'll talk a little bit about that, to de-risk these targets from patients. So what he did originally was to fish out T-cell receptors from checkpoint experienced individuals and then use a combinatorial yeast display system and overlapping peptide libraries to then create interaction interfaces that are based wholly on biology. So when you see the interaction and the cell is killed or lights up, then you begin, at that point, you begin to see, pull out those T-cell receptors and then identify those target antigens in the same screen. What we now use is AI in those libraries to now look at not just one peptide MHC complex or a series, but to look at across all HLA types and all peptides. So you're at the the point of the 10 to 15, 10 to the 16th complexity. This is something that is not achievable through normal computation, and really uses those large language models. We can then validate those with mass spec. And so using this sort of comprehensive approach, using AI to identify not just the on-target effect, the affinity and the efficacy for the target the new T-cell receptor is looking for, but also what is it not seeing? And as I affinity mature it, what are the off-tumor, on-target proteins that are now recognized by this T-cell receptor, looking both at framework interaction as well as the peptide interaction itself. So you start with this massive, massive proteome set and then work your way down. And again, this works across all HLA types and you can look at simultaneously many, many HLA types across about 10 to the 15th different peptides. And so that's really the pipeline of this trace approach. Using these AI models, in addition to the experimental data in yeast display, we're able to actually identify the sequences of the T-cell receptors, either AI-based protein design, TCRs, or even fished out from PD-1 responders, and then screen those through the yeast display library, predict what it's interacting with, identify the key off-targets that we may want to engineer out, engineer those T-cell receptors, and continue to iterate on this until you have something, that Goldilocks, which is that high affinity receptor that also has exquisite specificity. And you can do that, I'll touch on that in just a minute, you can do that by looking at how much interaction with the MHC complex, the framework, versus the discriminatory peptide touch points. And so this really has led to a huge set of libraries that Chris and others have generated now, and David, that are used to train these large language models. And it does predict decoding. And it's reaching the point now where we're trying to get to the point where you could simply show an MHC peptide complex and then predict or show a TCR and predict which MHC peptide complex it can come from. And that's kind of the ultimate goal that these guys want to get to. These are just a couple of examples that you can see with the, let's see, I don't know if we can do it, but that's fine. So the TCRs 1 and 4 are what we look for as the good, kind of the fantastic ones. These are highly specific and high affinity for the T-cell receptor. As you move your way back, the ones above at the top, these are sort of off-tumor, on-target specificity, and then others which are much, much less specific, but still high affinity for their targets. And so this is a single run. We can actually identify all of these different proteins in the context of multiple HLAs using this trace approach. And it's not only the fact that we can identify a really nice T-cell receptor. If you look, say, at the MAGE-A3, which now has known off-target effects, and then begin to screen that using the same platform, you very quickly identify kind of tightened reactivity, which of course was the kind of lethal side effect of that T-cell receptor, unfortunately, when we took it into the clinic. You can also identify where you could predict off-target could be. You might have a great T-cell receptor that may have skin reactivity for some proteins there, or you want to watch out for things like liver, heart, and brain. You can see that all of that as you begin to develop and engineer these and improve their affinity. And so really this approach, and we can also, again, this is the idea of increasing specificity. So if you look at benchmarks 1 and 2, these are clinical stage, one commercial and one clinical stage molecules that are out there. They have reactivity against their targets, but they also have cross-reactivity against other proteins, and that using this trace approach allows you to exquisitely engineer just against that wild-type target. And this really couldn't be done without the deep AI analysis. The other is, how do I improve specificity? And the way you do that is really, you take advantage of the peptide surface itself. So as the T-cell receptor dives down, the CDR3s are interrogating that. You need to have some level of framework interaction, but you want to also discriminate using multiple amino acids within the peptide. A single anchor means that you may get cross-reactivity against other proteins that are peptides that have that same amino acid in that position. And so what we can do is then engineer those TCRs so that they then don't just hit one point, but they read all the way across. And you see that here as well, that the same two TCRs, the benchmark TCRs, actually pivot really only on a single amino acid, whereas we can take other TCRs that hit the same protein and begin to engineer them so that they're much broader, and they can see that barcode that gives it much better specificity in the context of multiple HLA types. And then, so just touching on some of the products that came out of this, a 403 target the CRISPR company 3T Bioscience is off, and so that the 403 target is the one that they've recently identified as a novel tumor antigen. I'll show you, it's expressed in a number of different cancer types. It was identified through this TCR fishing approach and then subsequently improved through this trace interaction. And so it's expressed in colorectal cancer, both MSI and MSS colorectal cancer, at very high levels with very good penetrance across multiple subtypes. It's expressed in non-small cell lung cancer as well, breast cancer, and so we're quite excited about the ability to move this forward into the clinic. There's a possibility to move it in as a TCR, but I think we'll show you it's more likely going forward. What's going forward is a TCR mimetic. It's also expressed in renal cell carcinoma as well. When you look at this, it's high expressed across multiple subsets. We see this about 75% of RCC patients are expressing this, and when you look at that just from the Human Protein Atlas database, its expression is negatively correlated with overall survival. We think it's a great target in RCC, and as well as a number of other cancers. And so what we've developed really is this TCR mimetic molecule. This binds to, of course, CDR3, so it's like a T-cell engager, but at the same time uses a trace-derived, highly specific MHC peptide complex molecule that sees this peptide in the context of HLA-A2, and that clinical trial is pushing forward right now. So it's about to kick off in the next few weeks, and as it moves forward, we're really looking forward to escalating this. We'll start in colorectal cancer, but we're really going to move this forward into other areas. So just taking a look at when we think about AI-based protein design, it's not just the first stage, which is the ability to make T-cell receptors or TCR mimetics. It's really throughout the entire process. So it's the design phase where you're using things like RF diffusion and protein MPNN to actually build molecules that recognize your target, but you're then taking the harder step actually, which is saying of the 10 to the 15 peptide complexity across 10,000 different HLA types, how can I see whether or not I'm trying to get seeing off-tumor, on-target effects that are out there, and then using that then to optimize those and then to screen for off-target de-risking. So not just the identification, the orphanization, the de-orphanization of these receptors, but really creating them into true drug-like properties and then moving those into the clinic, and we're hugely excited about that. So these are things that I think were largely impossible prior to AI. You could probably, as Adaptimmune did, as others have done, as some amazing academic labs, Phil Greenberg and others have done in the past, they've used approaches to fish these out against known tumor antigens, things like PRAME. WT1's a great one out there, MAGE4, as you guys have seen. All of these are, it kind of limits your space. Now we're able to actually reach out, design TCRs against all of these new antigens and all of these different targets that are out there. But I think what we want to do is push this even further. So the programs that we're funding now and the RFAs that we've had come in, we're really around the big challenges in the TCR space. And I think from the commercialization standpoint and the real application in the clinic, the biggest challenge out there is the HLA barrier. Just the fact that I have to restrict to HLA-A201 is a real problem in moving this out there and when you actually start doing the numbers. And so the challenge we put to the network is break that barrier, use AI, use whatever methods you can to create molecules that can see these MHC peptide complexes across the barriers. I'll touch on one program, actually two different programs that are out there. The first really just use AI-based protein to kind of daisy chain these molecules. So these are sort of mini bodies built the same way I talked about before, which now can be built into single complexes, either in this case it's TCR-T, but also into TCR mimetics. So what this does is it'll see, for instance, the PRAME molecule in the context of A2, but it'll also see it in the context of A1 or A11 or A3, and these are really daisy-chained out there. The others are, there's other approaches that you guys can think about, which is this modular-based design where you could actually bring co-stimulation on board. We can get up to about 5 distinct MHC peptide binders into a single TCR molecule. It essentially represents an OR gate, so that if any of these are present, you can still get killing. This is being developed by groups here at the Hutch, but also at Stanford, and then there's a group at Oxford who's also pushing these forward and trying to move them as fast as possible into the clinic. This is kind of a brute force way to build these, but because of the small nature of these and the compact and stable nature of these AI-based proteins, you can begin to create these completely novel receptors to do something you couldn't do just with nature. I think another fantastic approach I'd love to highlight is the same kind of thing But now using AI-based protein work, this is again work from Chris Garcia's lab, but also Chris Klebanoff's lab at the Memorial Sloan Kettering, is to look at how the actual T-cell receptor interfaces with the MHC peptide complex, and then begin to engineer framework binders that will bind to not just one class 1 molecule, but super types of class 1 molecules. So by A2, A3, A11, because they're all kind of in the same superclass family. So shifting the framework and then being able to interrogate the surface of those receptors. Again, broadening now the visibility of that TCR for multiple HLA types. And so this is the big thing. So, you know, these kind of multimeric proteins, these different approaches allow you to overcome things like loss of heterozygosity, obviously allelic suppression, and create these large OR gates. I think this is the biggest outcome that we're going to be seeing in the next 5 years. We've already got early data in this space, and I think this is really going to change how we think about T-cell receptors and pushing them forward into the clinic. But this is not, you know, the only challenge. We've got a lot of challenges out there to the network, you know, huge hurdles that are out there in immunogenicity, off-target, you know, predictions. This is the big deal. TME suppression is still a big thing too. So bringing different kinds of co-stimulation or targeted cytokines with these is a big one. And we're tremendously excited by the work that's out there, foundational work that's out there, as you just heard, in the cancer vaccine space combined with these type of therapies. And so with that, I just want to acknowledge some of the things I talked about today, highlight some of the key papers that have come out just in the past few years that everyone should dive into, but really highlight the work of David Baker. And David's a part of the Parker Institute. His team is really phenomenal at producing these wonderful molecules. Chris Garcia's team, Chris and Aaron, both are dedicated to this idea of democratizing the availability and access to these kind of tools. And then the founding team, Marvin, Lan, and Stefan at 3T. I do also, so Aaron has a company called Araxa. This is basically an online service company. You can just go on, put your credit card in, and circle an area, and it'll design a protein against something. I always thought that was incredibly cool when you talk about democratization, because you can do it with coffee in one hand, so you know it's good. All right, so with that, I'd be happy to take any questions. Thank you.
I think, oh yeah, I think we're a bit over time, so maybe we'll save for the, no, but that was a phenomenal talk. Thank you so much.
Thank you very much.
All right, well, thank you. I think we're going to start a break a little, yeah, we are running a little bit behind, so thank you, everybody.
Thank you very much.
Thanks, everyone. Thank you.
Thank you very much.
Thanks.